Well, hello everyone. Well, as you know, we got they had the last exam, which was exam 3, and so that's been completed last night. I hope everybody's doing well. Hopefully you're being able to keep up with your family and stay healthy and all that. Hopefully you're being able to get us supplies of stuff. Our neighborhood is doing pretty good. Our neighbors are being very generous. We now have a supply of toilet paper that we can use for a long time. So hang in there, everything will be good. But I wouldn't recommend doing that. All right, well, let's go ahead and get started. So this chapter is basically on eukaryotic gene expression. And it's basically how we express our genes. And we are using this, we used the first chapter, which was over bacterial gene expression, to create the models necessary for us to explore mammalian gene expression, or eukaryotic gene expression more accurately. And so that's what this chapter is about. And so it's about gene regulation, eukaryotes, transcriptional and translational regulation as well. So let's go ahead and get started on this chapter. So if you're talking about gene expression, you're actually talking about the interaction of transcription factors with DNA to activate certain genes in certain areas of the genome. And that means that you're going to have some type of protein to DNA interaction, which is being represented in this picture. So as you can see, this is showing the major groove of the DNA. And as you can see, you have the alpha helical structures, or alpha turn helix structures, that are involved in this type of thing. So let's go into the introduction. So eukaryote organisms derive many benefits from regulating their genes. So for example, nutrient availability. They can respond to it much quicker without losing very much energy. They can respond to environmental stresses. For example, in plants and animals, multicellularity and more complex cell structures also demand a much greater level of gene regulation. So that means because of our multicellularity, we have to create different types of cells, meaning that we have to have a different level of gene regulation that goes beyond just turning genes on in response to environment. It has to be involved in how we create the different cells that allow us as a multicellular organism to respond to that environment. So this regulation is necessary to ensure that the expression of genes is in an accurate pattern during the various developments of the life cycle. So that means that genes that are supposed to create the arm or our limbs overall, they're supposed to be expressed in one area during gene expression, because initially when we form, those arms aren't there. They actually bud out the sides of our body and start to form. They also have to be regulated on how long they are influencing this particular structure. For example, if the cells in our arms should divide, if all the cells would have divided one more time, our arms would actually reach all the way down to the floor, and we wouldn't even have to bend over to tie our shoes. So there must be a mechanism in place to regulate that. So some genes are only expressed during embryonic stages, whereas others are only expressed in the adult. So there are genes we need to get through the embryonic stages, there are genes we need to get through the fetal stages, there are genes we need to get through the stages as an infant, teenage, all of that. They all are turned on at different times and turned off. Differences among cell types, meaning your cell structures. You have nerves, you have muscles, you have cells in the kidney, you have many different cells in the kidney that are involved in filtration. Cells look so different because gene regulation rather than difference in DNA content brings it about. What that means is we just didn't eliminate DNA for the vast majority of our cells. We didn't eliminate DNA to bring about those structures. We turned off certain genes and turned on certain genes to bring about those structures and to maintain those structures. So the complexity, I think you can understand where it is. So we have versus the bacterial cell, where the bacterial cell doesn't have a actual nucleus. In these particular cells, what we have here is we have to have regulation of transcription. We have to have regulation of the processing of the RNA being produced. The vast majority of the RNA molecules don't get out of the nucleus. They just don't form right, and they're eliminated and redone. We have to have translational control. How many of these messenger RNAs, how many polypeptides are we going to produce off of them? Do we have too many of those messenger RNAs? Do they eliminate those because we don't need that polypeptide? And in fact, there's even another whole level that we haven't even discussed, that we didn't approach in bacteria. There's what we call post-translational modifications. Some proteins have to be modified for them to actually be useful to the overall organism. Some of these have to have huge groups of carbohydrate structures added to these particular proteins to be utilized at the cell surface. Some of these carbohydrate structures act as a barrier to protect the cells. Some of them are used as sort of like a net to capture growth factors and to pull them next to the cell. Some of them are used just to interact with the matrix around the cell. So there's a lot of things that are necessary to do this, and they have to be built accordingly to what their function is going to be. And it all throughout this whole thing, including post-translational modifications, all of this is controlled at the DNA level. So, how do we bring about this regulation? Well, there are factors that are involved in this. We refer to these polypeptide factors as transcription factors. That means they are involved in transcription initially here. So they are proteins that influence the ability of RNA polymerase to transcribe a given gene. So they are going to be the ones that are going to identify which gene is going to be turned on. They're going to be the ones that bring in and promote the RNA polymerase to bind to that particular site. And they're going to be the ones that are going to regulate how often that transcription is going to occur. How often do we have the volume control and that type of thing. Okay? The first type, there are two main types. The first one that we've been looking at are the general transcription factors. They're required for binding of the RNA polymerase to the core promoter and its progression to the elongation stage, are necessary for just a small amount of transcription. So these are the guys that are coming in there and they open up the site for transcription. That means they have to go in there and they have to modify the nucleosome in some way to get that promoter out from the actual chromosome so that the transcription factors can bind to them and bring about the location of the RNA polymerase to that correct location on the actual promoter. Some of these particular ones that are involved in that, just finding them, we call them pioneer transcription factors. They literally are the ones that go out into this forest of DNA and find the right sites. Then we have the second level, which is regulatory transcription factors. They serve to regulate the rate of transcription of that target genes. They influence the ability of RNA polymerase to begin transcription of a particular gene. So they're pretty much turning on the activity, the volume control, all of that. They're there to determine that now we need that gene at this moment in time where this cell is located in this group of cells, in this particular group of tissue, in this particular group of the organ. So 2 to 3% of human genes encode for transcription factors in our body. That includes general transcription factors and regulatory transcription factors. Now, the regulatory transcription factors, if you remember, general transcription factors are looking at the core promoter. That's that portion that's 5' of that coding region for that particular gene. Well, regulatory transcription factors, they recognize what we call cis regulatory elements. That means these are fragments of the DNA that can be 5' upstream from the gene. They can be downstream from the gene. They can also be located within the gene itself. And so they are not exactly at the promoter, they are just on the same DNA fragment. These sequences are known as control elements, regulatory elements, or regulatory sequences. Okay, Now, keep in mind, they are what the actual transcription factors are binding to are what we call control elements, or regulatory sequences. The binding of the regulatory transcription factor to a control element affects the transcription of an associated gene. When it affects it, it means it affects either its level or it affects it in a way to where it's a temporal type of impact, where that gene is ready to go. It has all the other factors in place and it's just waiting for the last factor to come in and say, okay, now you're needed. Okay? It's like, for example, a surgical room. You have everybody in place ready for the surgery to begin, and the last thing you need to get surgery started is the actual surgeon to walk in the room and start it. So regulatory factors in this particular room, you've already got the basal transcription factors in there, the people who are caring for the patient while they're waiting in, and then you have some of them are regulatory factors, and they're preparing the patient for the surgeon. And then finally, the final regulatory factor is going to be the one that comes in, and that'll be the surgeon who starts the whole process. That's a fairly good analogy for you to understand what these things are. So a regulatory protein that increases the rate of transcription is termed an activator. The sequence it binds to is called an enhancer. That means it enhances transcription. A regular protein that decreases the rate of transcription is termed a repressor, and the sequence it binds to is called a silencer. So it's an interesting play of words, but regulatory factors can enhance transcription or they can inhibit transcription or repress transcription. The interesting thing about these things is that some regulatory factors that are involved in enhancing gene expression in other genes are actually inhibitors of gene expression in other genes. And in a way, that kind of fits with the concept that there's a reason why there's a muscle cell. Certain regulatory factors activated the genes that are necessary for muscle cells, but they turned off the genes, so they inhibited or repressed the genes that are necessary for creating nerves, that type of thing. So you can see how they can play a role in these systems. It gets confusing, yes, it's very complex. And we still don't know everything, we still don't understand everything about it when we go into this. Okay, so we're going to stop for a moment.